Hello, and welcome to this video in the series on Integrated Pest Management, dealing with disease control in cereals. We've all become familiar with a repeating cycle of new cereal varieties being introduced with good disease resistance, and then often within a few years that disease resistance breaking down when a new virulent strain emerges. And we've seen this pattern most clearly in the UK with yellow rust. When that happens, we have two choices. We can either stop growing the varieties or we can prop them up with fungicides. There's also a hidden cost because we often lose the breeding lines which share that resistance gene. So in this episode, we're going to look at what we can do to slow down the evolution of virulence so that resistance genes and varieties last longer. In this video on integrated pest management, we're going to take a look at the question, how to manage pathogen virulence. We need to be clear about a couple of concepts here about how pathogens can evolve to become more problematic. One option for them is to become more aggressive, which is that they obtain the ability to create faster epidemics more generally across a whole range of varieties, and or they gain the ability to cause disease in a wider range of environmental conditions. And we've seen this, for example, in recent decades in the UK in relation to potato late blight, and we've seen it globally with the evolution of adapted strains of yellow rust which are able to grow in warmer conditions and therefore cause epidemics in parts of the world that previously didn't have yellow rust problems. But the second evolutionary process, which is what we're focusing on here, is the ability of pathogens to evolve virulence and therefore be able to overcome specific resistance genes, in particular crop varieties. And the way they're able to do that is to mutate in a way that means the resistance genes in the plant aren't able to recognize that the plant is under attack by a pathogen spore, and therefore the resistance response doesn't kick in, or at least the resistance response is delayed. So in a normal resistance case, spore would land on the leaf surface, the plant would detect that it's there and block the infection. But if a virulent spore lands, we get what we see in the diagram here, which is an illustration of a sort of microscopic cross-section of a plant leaf where the spore has landed on the surface. It's been able to penetrate through the leaf surface and start to extract nutrients from the crop, thus damaging the leaf. And as we saw in the episode on how to make use of variety selection and particularly disease resistance, we looked at different types of disease resistance, one of which was what we termed major gene or qualitative resistance, which is usually obtained by a single resistance gene of large effect. And that's its Achilles heel because a single mutation in the pathogen can be enough to overcome a single resistance gene. And that can lead to new virulent pathogen strains that cause loss of effectiveness of that resistance. If we look at how that manifests itself in practice, we have two contrasting examples. On the left we have Septoria triticide blotch, and on the right we have yellow rust. In both cases, this is AHDB recommended list data showing the recommended list resistance rating against each disease on a scale of 1 to 9. What we can see on the left-hand side with Septoria tritici is that the resistance is generally of moderate effectiveness, and that's created by putting together usually a number of smaller genes of smaller effect, sort of minor genes of smaller effect. And the advantage of that is that the resistance tends to be more stable over time, and at worst what we see is a kind of gradual erosion of that resistance. On the right hand side, in contrast, we see that for yellow rust quite often the resistance is coded for by a major gene, and we can see a couple of examples here in this timeline, for example between 2009 and 2010 and around 2012, where a new yellow rust race has come along which is virulent and able to overcome the resistance of the varieties that contain that gene, causing a very sudden loss of effectiveness. So let's just try and understand the evolutionary process by which this occurs in order to understand what we can do about it. And to understand this, I'd like to just take a step back to what we looked at in the video on managing fungicide resistance, because there's a strong analogy between the evolution of fungicide resistance and the evolution of virulence. So what we looked at in that fungicide resistance video was the case where in the left-hand chart we have a population of strains of the pathogen that are sensitive to a fungicide, and then a newly emerged mutated resistant strain. And in the presence of the fungicide, that resistant strain clearly has a competitive advantage. The epidemic of that strain is going to proceed more rapidly than the sensitive strain, and that competitive advantage leads to what we see on the right, which is the fraction of the pathogen population which is fungicide resistant increasing through time, and at a certain point along that timeline we're going to have some loss of control that we were previously able to obtain from the fungicides. And the rate of selection for the resistant strain is determined by the difference in the per capita growth rate of the resistant and the sensitive strains and the time over which the fungicide is creating that selective pressure. Now, if we apply the same thinking to virulence, the same evolutionary process applies, but in this case we've got a set of avirulent strains to which the varieties are resistant, and then a newly emerged virulent strain that can overcome the host resistance gene and is therefore able to grow more rapidly on those varieties. As a result, the fraction of the pathogen population that is virulent increases through time, and at a certain point we lose control. Now, if you recall, we talked in the fungicide resistance case about the benefits of fungicide mixtures of different modes of action, and a similar principle applies in the case of virulence evolution. So if instead of mixing two fungicides of different modes of action, we mix in a variety two resistance genes or several resistance genes, on the left-hand side what we see is what would happen if we just had varieties with a single resistance gene A and a virulent strain that can overcome it. But if we have a second resistance gene B, which the pathogen is not virulent to, then that resistance gene B is going to slow the epidemic growth rate of both the virulent and avirulent strains to A, and therefore reduce the strength of the selection by reducing the difference in the per capita growth rates. And we see that in the bottom right diagram, that the rate of increase of the virulent fraction in the population is slowed. So what can we do to exploit this in practice? We have a number of options for how we could deploy a given number of available resistance genes. So the first of them is to stack multiple resistance genes into each variety, a process which is called pyramiding for reasons we'll see in the next slide. And we can stack together or pyramid major resistance genes. We can stack genes of smaller effect, the minor resistance genes. We could combine major and minor genes, or we could create multi-lines. So this is not commonly used, but it's possible to create a variety which is genetically uniform, but where different individuals within that variety contain different disease resistance genes. The other option is to put each gene into a different variety, and we could sow those varieties as a mixture or variety blend within a field, or we could sow varieties in different fields, as we commonly do, creating a mosaic of fields with different resistance genes in them. But typically we use all sorts of combinations of the options above, and we'll take a look at the consequences of that. So first of all, pyramiding resistance genes. If we look at the diagram on the left, we can see here 4 breeding lines of a cereal, and each of those 4 breeding lines contains a different resistance gene represented by the different shaped symbols. If we cross pairs of those 4 breeding lines, and we end up now with 2 breeding lines, each of which contains 2 genes for resistance, provided we select the offspring of the cross that contain both of the genes available from their parents. If we then cross those 2 offspring, we can end up with offspring which contain all 4 of the original resistance genes stacked or pyramided into a single variety, and it's termed pyramiding because of the shape of the diagram on the left. So the consequences of doing this are several. The first is that if we accumulate together or pyramid a whole bunch of minor genes of small effect, then we improve the disease control, and we see this particularly in the improvements that have been made in controlling Septoria triticide through plant breeding. By pyramiding resistance genes, we also make it more difficult for the pathogen to overcome the resistance of the variety, because it's going to need multiple mutations to happen before it's going to be able to reproduce successfully on that variety. So it's clear that pyramiding extends the life of resistant varieties. What's somewhat less clear is whether pyramiding actually extends the life of each individual resistance gene and how much benefit we get from it. Partly because although we have the kind of mixture effect of pyramiding, which is a benefit, we're also creating evolution, or the pressure for evolution, against multiple genes all at once. However, the modelling evidence on this and the general consensus is that pyramiding is useful for reducing virulence selection. And the final advantage of pyramiding is that if we combine major and minor genes into a variety, if the major gene breaks due to a virulent variety, we don't get a sudden catastrophic drop to extreme susceptibility, because the minor genes are still effective and helping to limit disease. So now let's take a look at how we might deploy varieties that have either got single genes or pyramids within them. So let's take a look first of all on the left. This is a kind of illustration of a single field. We could choose to take 2 or more varieties, mix up all the seed and sow the varieties all into one field as a variety mixture or blend. Now we can see the consequences of that in the diagram on the right hand side. So if we sow a pure stand of a variety which the resistance has been overcome, then any spores produced by a virulent strain on any of those plants will land on neighbouring plants, and those plants will also be susceptible, and the epidemic is going to run away very quickly. On the right hand side of the diagram we see an example of a variety blend where the blue plants still contain genes which are effective against the strain which can overcome the resistance in the red plants. So now what we see is that spores produced on the susceptible plant are going to move around in the crop, and probably about half of them are going to land on a plant that they can't infect, and that's going to slow down the rate of epidemic development. And that also has the effect of reducing the selection of virulence, because it reduces the difference in the per capita growth rate between the virulent and the avirulent strain. So this is again analogous to the fungicide resistance case. There are 2 differences here though. One is that the fraction of plants resistant in the blend has a key effect on the evolution of virulence. And secondly, the time over which the selection happens is determined by the time over which the disease resistance gene is expressed in the plant, and the plant is subjected to infection pressure. And we'll see the implications of this a bit later on. The other option is that we take varieties, some of which will be susceptible, some of which will be more resistant, and plant them in separate fields. So in principle the same thing applies, that spores produced from fields of the susceptible variety, a fair proportion of them might land on fields of the resistant variety, and this will help to slow down the global epidemic. But there's quite a limitation on this in practice, because the majority of spores that are produced in a field are circulating within that same field, and only a proportion of them blow away and land in neighbouring fields. And the general consensus is that this effect makes a mosaic much less effective than a variety mixture or blend. So the other thing we could do is illustrated by these diagrams here. On the left we have the figure that we saw before with a virulent strain and an avirulent strain, leading to an increase in the fraction virulent through time. But if we imagine we applied a fungicide program to that field, then the fungicide should be effective against both the strain that's virulent and the strain that's avirulent, and will reduce the growth rate of both. And that has the effect of reducing the difference in the growth rate between the strains, and therefore reducing the rate of selection for virulence. And we can see that in the bottom right diagram, where the fraction virulent is growing more slowly. Now, the limitation on this is that the benefit of having the fungicide treatment only occurs during the period when the fungicide is slowing the epidemic and the resistance gene is effective. And we can see how that plays out in the next slide. So if we imagine this as a timeline for a season of a cereal variety, we can see, if we take the case of Septoria tritici, that the pathogen can be reproducing pretty much from the point of crop emergence right the way through to beyond when the crop has senesced and pre-harvest. And if we have a resistance gene which is expressed right from the seedling stage, then that disease resistance expression can also occur for quite a long period through the crop season, right through until the crop starts to naturally senesce. But the fungicide protection that we typically apply might start somewhere around growth stage 31 at first node and extend through to, say, 3 weeks after the last spray, which is typically sometime around ear emergence. Now, when we account for the fact that the rate of pathogen reproduction is dependent on temperature, we need to look at this in terms of thermal time. And when we look at that, what we can see is that the period of fungicide protection is typically somewhere less than half of the period during which the disease resistance gene is being expressed. So the selection pressure for virulence is expressed for perhaps most of the season, about 3,000 degree days, whereas the benefit of fungicide reducing that selection is only occurring for perhaps less than half of that time, which limits the benefit to a considerable extent. And we definitely should not be thinking about extending the period of fungicide protection in order to protect disease resistance, because the disbenefit of that to fungicide resistance evolution would far outweigh the benefit to virulence evolution. So what can we conclude? Pyrimidine resistance genes increases the durability of disease control, and marker assistance is helping plant breeders achieve that to a greater extent, and we're seeing the success of that coming through in modern varieties. Pyrimidine major resistance genes with minor genes can help reduce the sudden loss of control when the major gene is overcome, and again, markers can help with this because visually in a breeding program the effect of minor genes is masked by the effect of the major gene or genes. Variety mixtures or blends can reduce the epidemic growth rate, and that has the side benefit of reducing the selection for virulence. And variety mixtures are probably more effective in this regard than sowing different varieties in separate fields. And we also have some potential for mutual protection with variety disease resistance slowing fungicide resistance and fungicides slowing virulence. And if you'd like to know more on this subject, there's plenty of literature and guidance to look at.